The next pattern of cell death we'll be talking about is necrosis. Necrosis. So what is necrosis? Necrosis is simply the morphologic spectrum that follow pathologic cell death. Okay, so it's a spectrum, okay, and of course it is pathological. So unlike apoptosis, necrosis is always pathological, okay, and it is characterized by cell membrane damage, and this membrane can be the plasma membrane, or other organellar membrane. Okay, leakage of cellular contents out of the cell, and of course inflammation. Okay, the common causes of necrosis include oxygen deprivation, that is hypoxia, okay, certain chemical and even physical agents that will induce severe injury to the cell, and of course eventually lead to cell death. Now, when necrosis happens, when cell membranes are damaged, okay, contents of the cell leak out of the cell, and when these contents leak out of the cell, they elicit an inflammatory response. Now, these contents of the cell have been called damage associated molecular patterns. Okay, which is what we call DAMPs. Damage associated molecular patterns, and they are recognized by the immune system. These constituents are supposed to be confined within the cell, but the moment they appear outside of the cell, it becomes a sign that something wrong is going on. Just for example, if I come into this place and I see blood everywhere, I will know exactly that something wrong has happened. That is exactly, or that is an idea of how these molecules are perceived, okay? And examples of these molecules that have been termed the damage-associated molecular patterns include uric acid, ATP, DNA molecules, and changing concentrations of potassium ion. Okay, these things and several others are damage-associated molecular patterns, and of course they are recognized by the immune system. Okay, now there are characteristics of necrosis, okay, that we can see under the microscope. Necrotic cells have an increased eosinophilia, meaning that they are more pinkish, okay, and that is usually because of digestion of cytoplasmic RNA. Okay, and if the cell is still retaining its structure, usually the cell is swollen, okay, with blebs, with cytoplasmic blebs. Okay, so these are some of the things we see in the cells. There are also nuclear changes in necrosis, one of which is karyolysis, where the nucleus simply disintegrates and disappears because of digestion. We have pycnosis, where the cell shrinks, and pycnosis also happens in apoptosis, okay, where the cell shrinks. And finally we have karyorrhexis, okay, where the cell fragments, where the nucleus, fragments, okay, So these are nuclear changes, karyolysis, pycnosis, and karyorrhexis, that we see in necrosis. There are certain ultrastructural changes also that may not be seen under the light microscope but can be seen under electronic microscope. We see in that case, we see cell membrane fragmentation. The cell membrane will have spaces in between it. We will see organella edema, particularly the mitochondrium. Okay, we will see myelin figures, okay, within the cell. Okay, and these myelin figures are as a result of disintegration of cell membrane phospholipids. Okay, we will also see certain amorphous densities or amorphous structures, okay, that are evidence of protein denaturation. These are some of the changes that we see ultrastructurally in necrosis. Now, there are different forms of necrosis. These morphologic patterns include coagulative necrosis, liquefactive necrosis, fibrinoid necrosis, caseous necrosis, fat necrosis, and of course gangrene, which is not particularly a pattern of necrosis but has been traditionally ingrained in pathology. Okay, so coagulative necrosis is simply the form of necrosis in which the cell and the tissue retain their architecture. Okay, so there's retention of cellular and tissue architecture. Retention of architecture in coagulative necrosis. And the reason is because the injury that incited the necrosis not only denatured the cellular content, but also denatured the enzymes so that they cannot digest the cell or the cellular contents. Okay, so that's what happens in coagulative necrosis. And usually it takes some time, and then shortly after, inflammatory cells come in and release enzymes to digest these constituents. And one common cause of coagulative necrosis is ischemia. Okay, and a focus of coagulative necrosis as a result of ischemia is called an infarct. Okay, now in liquefactive necrosis, there is digestion of cellular constituents so that the necrotic tissue will have a viscous appearance. One very simple example that we can all relate with is pus, okay, where there is digestion, okay, of cells, and of course formation of these viscous liquid material. That is basically what liquefactive necrosis is. And liquefactive necrosis in the brain, in the central nervous, in the brain, is usually always liquefactive. There is no coagulative necrosis happening in the brain. And this is because the brain has a lot of enzymes that can easily digest tissue. Okay, so liquefactive necrosis. In fibrinoid necrosis usually happens in blood vessels as a result of immune reaction, usually where there are depositions of antigen-antibody complex in the walls of blood vessels. We see fibrinoid necrosis, and it is deeply eosinophilic. So it's a deeply eosinophilic necrotic area around blood vessels with some inflammatory infiltration. That is what fibrinoid necrosis is. Now, caseous necrosis is commonly seen in diseases like tuberculosis, okay, where the necrotic area will resemble cheese. So it's a cheese-like appearance, and that is why it is called caseous necrosis. Histologically, there is a necrotic area, okay, with inflammatory cells, and some of these inflammatory cells, macrophages, have fused to form giant cells. That kind of histologic focus is what is called a granuloma, okay? But caseous necrosis looks cheese-like. The next thing we'll be talking about is fat necrosis. In fat necrosis, it is usually as a result of release of enzymes that digest fatty tissue, and it is very commonly seen in acute pancreatitis, where pancreatic enzymes are released and begin to digest tissues. In such cases, the fatty acids, okay, the release of these phospholipids can bind to calcium and form certain areas of calcifications that look whitish like chalk, okay? And that's basically what fat necrosis is. Histologically, we will see necrotic fat infiltrated by inflammatory cells. And then when we talk about gangrene, we simply mean a necrosis of part of the limbs. Okay, that is what gangrene is. It's not particularly a morphologic pattern, but because it has come to stay in pathology, and we will not talk about necrosis without talking about gangrene. And gangrene can be wet gangrene, okay, when there is superimposed infection and digestion of the cells, or it can be dry gangrene where it is as a result of coagulative necrosis. Okay, so these are the morphologic patterns or the morphologic spectrum of necrosis. Like I said earlier, necrosis is always pathological. Apoptosis can be physiologic or pathological. In apoptosis, the fragments are membrane bound. In necrosis, they are not membrane bound. Cellular constituents leak out. Okay? In apoptosis, there is no inflammation. But in necrosis, because of those cellular contents that have leaked out, there is inflammation. Apoptosis does not necessarily have a morphologic architecture apart from the apoptotic bodies. But of course, necrosis has a morphologic architecture histologically. Okay, and histologically, necrosis will appear as a pale eosinophilic, amorphous density. Okay, density. That is what necrosis looks like. Now, the necrotic tissue are usually, digested away by enzymes or even phagocytosed by macrophages. Okay, so with that, we have come to the end of the summary of necrosis. I hope you enjoyed the video. In our subsequent video, we'll be talking about pathologic calcifications and intracellular accumulations as we draw the curtain on cellular pathology, an overview of cellular pathology. Please, I would like you to subscribe if you have not subscribed, like the videos, make comments, and give us suggestions of topics that you would like us to talk about. Thank you very much. I'll see you in the next one.